All right, good evening, guys. Welcome to biology class. Okay, we started a series in the previous weeks on cell theory, everything about cell theory, cell properties, the properties of plant cells, the properties of animal cells, and all that. Okay, differences. We're going to look at all that today. So we started by looking at the features, the characteristics of the properties, or rather, the components of a cell, their analysis. So far, we've looked at a nucleus, we've looked at cytoplasm, we've looked at mitochondria, and all that. Okay, now we're done with this. Next class, we're going to be looking at cell division, a meiotic cell division, and mitotic cell division. All right. So I want to put finishing touches to the remaining components of a cell. All right. So today we'll continuing with endoplasmic reticulum. All right. For those that just joined the group newly, and you missed out on the previous biology videos. All right. You do let me know. I'll send the YouTube link to the group so that we can have access to those videos. Okay. So today we're looking at endoplasmic reticulum. All right. So the endoplasmic reticulum is an organelle found in eukaryotic cells only. Yeah, for I think yes, some weeks now I'm looking at prokaryotic cells and eukaryotic cells, and I made us understand that prokaryotic cells they are cells without a true nucleus. They are cells without a true nucleus. Why eukaryotic cells are cells that they have nucleus. They have true nucleus, true nucleus. All right. So the endoplasmic reticulum is found in eukaryotic cells only. So that's one of the differences. Take notes, the jam question. That's one of the differences between prokaryotic cells and eukaryotic cells. Okay. So the endoplasmic reticulum has a double membrane consisting of a network of hollow tubes. Okay. Flattened sheets and round sacs. Okay, so this is just a description of the endoplasmic reticulum. You can see the diagram, the schematic diagram below. Okay, so these flattened hollow folds and sacs are called cisternae. The endoplasmic reticulum is located in the cytoplasm. Let's take note. It is located in the cytoplasm and it is connected to the nuclear envelope. Okay, so there are two types of endoplasmic reticulum. We have the smooth endoplasmic reticulum and we have the rough endoplasmic reticulum. So for the smooth endoplasmic reticulum, it does not have any ribosomes attached to it. It does not have any ribosomes attached to it. And also it is involved in the synthesis of lipids, including oils, phospholipids, and steroids. It is also responsible for metabolism of carbohydrates, regulation of calcium concentration, and detoxification of drugs. That's all on the smooth endoplasmic reticulum. Now for the rough endoplasmic reticulum, it is covered with ribosomes, giving the endoplasmic reticulum its rough appearance. Okay, you can see the diagrams below. It is also responsible for protein synthesis and plays a role in membrane production. The folds present in the membrane increase the surface area, allowing more ribosomes to be present on the endoplasmic reticulum and thereby allowing greater protein production. While for the former, we'll see the synthesis of lipids, phospholipids, and steroids. While for the latter, for the rough endoplasmic reticulum, we'll see the synthesis of protein and the production of protein. So we have the schematic diagram below. You can see the smooth endoplasmic reticulum, the rough endoplasmic reticulum. Okay, so that's that endoplasmic reticulum. All right, next is ribosomes. Okay. And when I'm done with all these, I think the latter end of this a video recording, we'll see a summary of everything we've looked at, so that At least if you missed the previous classes, you can follow up by having that summary with you. All right. So ribosomes are composed of RNA, ribonucleic acid, and protein. Okay, they occur in the cytoplasm. You can see the endoplasmic reticulum occurs in the cytoplasm. The ribosomes also occur in the cytoplasm, and they are the sites where protein synthesis, protein production occurs. That means ribosomes, the key function of ribosomes is for the synthesis or the synthesizing or production of protein synthesis. Ribosomes may occur singly in the cytoplasm or in groups, or may be attached to the endoplasmic reticulum, thus forming the rough endoplasmic reticulum. Ribosomes are important for protein production. I've said that earlier on, together with a structure known as messenger RNA, a type of nucleic acid. No, we have DNA, we have RNA. RNA is a type of nucleic acid. DNA, deoxyribonucleic acid, is also a type of nucleic acid. So together with a structure known as a messenger RNA, ribosomes form a structure known as a polyribosome, which is important in protein synthesis, Okay, we see the diagram, we see the free ribosome, then we see polyribosome. Uh, um, when ribosome is connected with the messenger RNA, you have a polyribosome. So that is all on ribosome. A summary, a simple analysis on ribosome. So next we're looking at this Golgi body. All right. The Golgi body is found near the nucleus and endoplasmic reticulum. The Golgi body is found near the nucleus and endoplasmic reticulum. I think in the first diagram I gave all that on the first slide. So it is found near the nucleus and endoplasmic reticulum. If I have access to a cell diagram, you see all this there. I think I dropped the diagram in the previous classes. I dropped the diagram. All right. So the Golgi body consists of a stack of flat membrane. Okay. It consists of a stack of flat membrane bound sacs called cisternae. Okay. The cisternae within the Golgi body consists of enzymes which modify the packaged products of the Golgi body. Okay, so see the diagram there, that's the Golgi bodies. See the diagram right there. Now, in fact, the Golgi body was discovered by the Italian physician Camillo Golgi. It was one of the first organelles to be discovered and described in detail because its large size made it easier to observe. Functions of the Golgi body. It is important for proteins to be transported from where they are synthesized to where they are required in the cell. So transportation of proteins from the point of synthesis to the point of requirement in the cell. Okay, from the point where they are synthesized to the point where they are required in the cell. So the transportation of that is a function of the Golgi body. All right, the organelle responsible for this is the Golgi body. I've said that the Golgi body is the sorting organelle of the cell. The sorting deals with arrangement, yes, classification, the sorting organelle of the cell. Proteins are transported from the rough endoplasmic reticulum to the Golgi. In the Golgi, proteins are modified and packaged into vesicle. They are modified and they are packaged into vesicle. The Golgi body, therefore, receives proteins made in one location in the cell and transfers these, can you see, and transfers these to another location within the cell where they are required. I've laid emphasis on this before. All right, so for this reason, the Golgi body can be considered to be the post office of the cell. That they are called the post office of the cell. So that's all on Golgi body. Okay, quickly the next one. I think we're coming to a close on this aspect. vesicles and lysosomes. Vesicles and lysosomes. All right. So vesicles are small membrane-bound spherical sacs which facilitate the metabolism, transport, and storage of molecules. Many vesicles are made in the Golgi body and the endoplasmic reticulum, or they are made from parts of the cell membrane. Vesicles can be classified according to their contents and function. Transport vesicles also transport molecules within the cell. So that's all on vesicles, okay? Now for lysosomes, they are formed by the Golgi body and contain powerful digestive enzymes that can potentially digest the cell. All right. So lysosomes are formed by the Golgi body or the endoplasmic reticulum. So these powerful enzymes can digest cell structures and food molecules such as carbohydrates and proteins, and they are abundant in animal cells that ingest food through food vacuoles. So when a cell dies, the lysosomes releases its enzymes and digests the cell. Digests the cell. That is the function of the lysosomes, for digesting cell structures and food molecules. All right. So the vacuoles, quickly. The vacuoles are membrane bound. Can you see all these things? They are membrane bound. So they can ask you, for example, which of the components of a cell are not membrane bound? I've mentioned vacuoles, I've mentioned vesicles, I've mentioned lysosomes, I've mentioned endoplasmic reticulum, I've mentioned Golgi bodies. These components of the cell, they are membrane bound. They are bounded by membrane, by cell membrane. So vacuoles are membrane bound, fluid filled organelles that occur in the cytoplasm of most plant cells, but are very small or completely absent from animal cells. They are completely absent from animal cells. Plant cells generally have one large vacuole that takes up most of the cell's volume. A selectively permeable membrane called the tonoplast surround the vacuole. The vacuole contains cell sap because they are present in animal cells. So they contain cell sap. Sorry, they are present in plant cells rather. They contain cell sap, which is a liquid consisting of water, mineral salts, sugar, and amino acids. So vacuoles are found in plant cells and they are absent, totally absent in animal cells. So what are the functions of the vacuole? The vacuole plays an important role in digestion and excretion of cellular waste and storage of water and organic and inorganic substances. So they play an important role in digestion, in excretion, and in storage of water, storage of organic substances, and storage of inorganic substances. Number two, the vacuole takes in and releases water by osmosis. The vacuole takes in and releases water by osmosis in response to changes in the cytoplasm as well as in the environment around the cell. So it takes in and releases water by osmosis in response to the changes in the cytoplasm as well as the environment around the cell. Also, the vacuole is also responsible for maintaining the shape of plant cells. Since it is found in plant cells, it is responsible for maintaining the shape and the structure of plant cells. When the cell is full of water, the vacuole exerts pressure outside, pushing the cell membrane against the cell wall. And this pressure is called the Tugor pressure. The Tugor pressure is the pressure exerted by the vacuole, exerted externally or outwards by the vacuole, pushing the cell membrane against the cell wall. Tugor pressure. And also we see that if there is not sufficient water, pressure exerted by the vacuole, which is the Tugor pressure, the pressure exerted by the vacuole is reduced and the cells become flaccid, causing the plant to wilt. So if there's not sufficient water, the pressure exerted by the vacuole is reduced and the cells become flaccid, causing the plant to wilt. Okay, yes, I think the two last ones I have here, centrioles and plastids. All right, animal cells contain a special organelle called a centriole. Just the way you have vacuoles and plant cells only and not in animal cells, then you have centrioles in animal cells and not present in plant cells. So the centriole is a cylindrical tube-like structure that is composed of 9 microtubules arranged in a very particular pattern. All right, so two centrioles arranged perpendicular to each other are referred to as a centrosome. They are referred to as a centrosome. Two centroles arranged perpendicularly to each other are referred to as a centrosome. The centrosome plays a very important role in cell division. Yes, I think that's what we're going to continue our next class. Cell division, mitotic cell division, meiotic cell division. So the centrosome plays an important role in cell division. The centrioles are responsible for organizing the microtubules that position the chromosomes in the correct location during cell division. Okay, yeah, you will learn more about this and we'll start cell division. Okay, now lastly, plastids. Plastids are organelles found only in plant cells. There are three types of plastids. You have the leucoplasts. They are white plastids found in the roots, the chloroplasts, the green colored plastids found in plants and algae, the chromoplasts. They contain red, orange, or yellow pigments, and they are common in ripening fruits, flowers, or autumn leaves. So, lastly, the color of plant flowers, such as an orchid, is controlled by a specialized organelle in a cell known as the chromoplast. So these are the all components of a cell we've looked at for the plant cell and also for the animal cell. Okay, now we have looked at the basic functions, structures of the organelles in a cell. We want to look at the key differences. These are the key differences, the differences between plant and animal cells. They're more than these. They're more than these. We just picked out the key differences between plant cells and animal cells. The first one, we see that plant cells don't contain plastids. Yes, that's the last one we look at. Plastids are only found in animal cells. Almost all animal cells don't contain plastids. Plastids are only found in plant cells. Almost all plant cells contain plastids, such as chloroplast, chromoplast, and leucoplast. Right. Number two, animal cells don't have cell wall. Plant cells have a rigid cellulose cell wall in addition to the cell membrane. Number three, animal cells contain centrioles. Plant cells do not contain centrioles. Can you see? Animal cells contain centrioles. Plant cells do not contain centrioles. Rather, they contain vacuoles. Vacuoles. All right. The fourth one. Animals do not have plasmodesmata or pits. We looked at that in the previous class. Plasmodesmata or pits. While plant cells, they contain plasmodesmata and they also contain pits. Number five, few vacuoles, if any. So animal cells have a scantily number of vacuoles, while plant cells, they have a large central vacuole filled with cell sap in mature cells. Next, the nucleus is generally found at the center of the cytoplasm in animal cells. The nucleus is generally found at the center of the cytoplasm in animal cells, while in plant cells, the nucleus is found near the edge of the cell. Near the edge of the cell. Check out the two diagrams. You will see this analysis I'm talking about. Then lastly, no intercellular spaces found between the cells in animal cell, while large intercellular air spaces found between some cells in plant cells. Okay, summary, we look at the discovery of cells. All living organisms are made of cells. Cells are very small, therefore magnifying instruments such as lenses and microscopes are used to view them. All right, by using a light microscope, the simple features of cell can be studied. So these are the summary of cell theory. Robert Hooke used a light microscope to examine non-living cork cells. Anthony van Leeuwenhoek was the first person to observe living cells using a microscope. Okay, so the development of cell theory was from the study of microscopic cells. Now, the summary on the cell structure and function. We see that all cells have the same basic structure. They are all surrounded by a cell membrane and they contain cytoplasm and organelles. All cells contain cytoplasm, all cells contain organelles. Cells have different sizes, shapes, and structures in order to carry out specialized functions. All right. The cell membrane is made up of phospholipids and proteins and also controls substances which move in and out of the cell. The structure of the cell membrane is referred to as the fluid mosaic model. The nucleus is made up of a nuclear membrane with nucleopores, chromatin material, and the nucleolus inside the nucleoplasm. Mitochondria, which is the powerhouse of the cell, the energy generator of the cell, release chemical potential energy, ATP, adenosine triphosphate, for the cell during cellular respiration. Ribosomes are important for protein production or protein synthesis. Cytoplasm is used for storage and circulation of various materials. Endoplasmic reticulum transports substances from one part of the cell to another. The Golgi body modifies, secretes, packages, and distributes various organic molecules such as protein and lipids around the cell. Vacuoles are used for storage. In plant cells, these are large, while in animal cells, if present, are very small. Lysosomes are mainly found in animal cells. Centrioles are only found in animal cells. The cell wall is found only in plant cells and is made up of cellulose. The cell wall gives the plants shape, support, and protection. Plastids are found only in plant cells. There are three types of plastids. You have the chloroplasts, which contain chlorophyll, and their function is the production of food by photosynthesis. We have the chromoplasts, which give colors to fruits and flowers, and we have leucoplasts, which are white and are used mainly for starch storage. So in our next class, we'll be looking at cell division. Thank you very much.